Doing well. In the last class, I have explained you about the different organs that forms our digestive system. The different organs that are found in the alimentary canal or digestive tract. So up till we were discussing about that organs. Now in today's class, let us talk about the different glands that helps in carrying out the process of digestion, and that glands are called as digestive glands. So there are 5 major type of digestive glands that are found in the human body. The first one is salivary gland. When we talk about the salivary glands, they are present in 3 pairs. 3 pairs of salivary glands are present in the buccal cavity. So that 3 pairs includes parotid gland, submaxillary, or we can say submandibular gland, sublingual gland. All 3 glands are present in the buccal cavity, and these glands are made up of some specialized cells, like they are made up of some serous and mucus cells, and these cells are responsible to secrete saliva. Is secreted by these 3 pairs of salivary glands, and this saliva contains many enzymes, water, salts. All these are the different substances that we can see in the saliva. We'll see what are the different enzymes that are responsible to mix up the food with the saliva and to break down the carbohydrate which is present in the food product. So this saliva contains some specialized enzymes, and that enzymes are ptyalin, we can say, and this is also called as salivary amylase. So salivary amylase is an enzyme that is present in the saliva which is secreted by our salivary glands, and one more enzyme is present in the saliva, and that enzyme is lysozyme. So whenever we heard the name of an enzyme, lysozyme, we need to remember its function, and the function of the lysozyme is to kill the bacteria. So lysozyme is an enzyme that is responsible to kill the bacteria, whereas salivary amylase is an enzyme that secretes alpha amylase compound in it, which helps in the process of digestion. So this is about the salivary glands, and the pH of salivary glands will always be 6.8, or it will be approximately 8. Next glands are gastric glands. The pH of gastric glands ranges from 0.9 to 1.8. And when we talk about gastric glands, then again the gastric glands are of 3 types. They are cardiac glands, pyloric glands, and fundic glands. All these glands are present that are responsible to secrete some substances, and the substances which gets secreted by these glands together form a juice called gastric juice, and this gastric juice again helps in the process of digestion and absorption of our food. The next one is intestinal glands. All these are the different digestive glands that helps in carrying out the process of digestion and also the process of absorption. So next one is intestinal glands. Intestinal glands. In the intestine, 2 main glands are present, and that glands are Brunner's gland and crypts of Lieberkühn. These 2 glands are responsible to secrete some peptidases and also to secrete some disaccharides. Peptidases like tripeptides, dipeptides, and amino peptides. All these are the different substances that are going to be secreted by these glands. And disaccharides are the sugar molecules like sucrase, maltase, lactase. The different sugar molecules are secreted by these glands. And these intestinal glands are lined by some specialized cells which we call Paneth cells. These Paneth cells contain an enzyme, and that enzyme is lysozyme again. Just before I told you that salivary glands also secretes lysozyme, which is responsible to kill the bacteria. Again, Paneth cell also contain an enzyme that is lysozyme, which helps in killing the bacteria and prevents the destruction in our stomach. This is how the different glands are present. The different secretions, they are going to be secreted, and that secretions helps in the process of digestion. it also helps in the process of metabolism of the different nutrients. So in such a way, the breakdown of different complex substances takes place by the secretion of different glands, and we call that glands as digestive glands. Besides this, besides salivary glands, gastric glands and intestinal glands, liver is an organ that also acts as the largest gland in the body. So liver is the largest gland of the human body, which helps in the different processes. So firstly, we'll see what are the different glands that are present in the liver and what is the structure of liver. As I told you that liver is the largest gland and it secretes the digestive juice called bile, which is very, very important in the process of digestion. Now we'll see the different ducts, the different glands that are present in the liver, how the different ducts are connected with each other and how they are going to secrete the digestive juices. Let us see. Firstly, when you see the structure of a liver, it is present below the diaphragm, and the liver, it contains 4 lobes. It consists of 4 lobes, but the 2 lobes are important one. The first lobe is left hepatic lobe and the second one is right hepatic lobe, and this liver is situated at the left side of the stomach cavity. The right side you can find the presence of stomach, but on the left hand side you can see the presence of an organ which acts as the largest gland, that is liver. And based on the position like left and right, this shows the presence of 2 main ducts, and the first, this one is left hepatic duct we can say, and this one is right hepatic duct. When both these ducts, left hepatic duct and right hepatic duct, join, then they form one common duct, and we call that duct as common hepatic duct. One balloon-like structure is also present in the left hepatic lobe of our liver, and this balloon-like structure is called as gallbladder. Gallbladder also stores the digestive juice called bile. It also has a gland that helps in carrying that bile to the different organs for the process of digestion. So the gallbladder also contains a duct, and this duct is called as cystic duct. Now this one is left hepatic duct, this one is our right hepatic duct. Both of them joining and it is forming a common hepatic duct. Now the cystic duct of the gallbladder joins this common hepatic duct, and 3 ducts, left hepatic duct, cystic duct and right hepatic duct. When these 3 joins together and when they form a common duct, then that duct is named as common bile duct, or we can write CBD, common bile duct. These 3 ducts join together and forms the common bile duct. So the bile which is stored in the gallbladder can be easily transferred through this duct. Now this common bile duct, it opens into the duodenum, which is the first part of the small intestine, but that opening is going to be controlled by a sphincter. So here you can find one sphincter which is present at the end of the common bile duct, and that sphincter is called as sphincter of Boyden. The sphincter controls the opening and closing of this common bile duct into the duodenum. Now this sphincter of Boyden can also be opened in one more gland, and that gland is present in another organ, that is pancreas. So in the pancreas, one duct is present that takes up the secretion like bile from the liver and gallbladder through the common bile duct, and that duct which is found in the pancreas is called as duct of Wilson. So this duct is going to collect the bile through a duct which is called as common bile duct. Now at the end of this duct some swelling is present. That swelling part is called as ampulla of Vater, and at the end you can see the presence of sphincter again. This duct also shows the presence of sphincter, and we call this sphincter as sphincter of Oddi, or we can pronounce Oddi also. So sphincter of Oddi is present, and this sphincter opens into the first part of the small intestine, that is in the duodenum. So this is how the bile which is secreted by the liver and gallbladder collects with the help of different ducts, and it is going to be transferred into the duodenum, and the pancreatic juices also help in the process of digestion. So these pancreatic juices are also be collected by a duct called duct of Wilson, which is present in the pancreas, and that pancreatic juice and the bile juice is going to be transferred into the first part of the small intestine, that is in the duodenum, for the process of digestion. So this is how all the different digestive glands are interconnected, and they are helping in transferring the different juices that are very, very important for carrying out the process of digestion as well as for the process of absorption. I am going to give a quick recap of this. Liver shows two main lobes. This one is left hepatic lobe and this one is right hepatic lobe. In the left hepatic lobe, a duct is present which is called as left hepatic duct. In the right hepatic lobe, one more duct is present, and this duct is called as right hepatic duct. Now, left and right hepatic duct, both of them join and they form a common hepatic duct. This common hepatic duct is present now, and to this common hepatic duct, one more duct joins, and that duct is of gallbladder. So in the gallbladder, cystic duct is present. Now when this cystic duct attaches to this common hepatic duct, then they form a common passage, and that common passage is called as common bile duct. This common bile duct at the end contains a sphincter. That sphincter is called as sphincter of Boden, and this sphincter opens into the duodenum of the small intestine. One more duct is present in the other organ, that is in the pancreas, and the duct which is present in the pancreas is called as duct of Wilson. This duct shows some swelling at the end of it, and that swelling part is called as ampulla of Vater, and ampulla of Vater, and at the end of this duct again you can find the presence of a sphincter, and we call this sphincter as sphincter of Oddi. So all the different ducts are joining together and they are opening in the duodenum to transfer their secretions like bile juice and pancreatic juice. So just before we have learned that how the different glands are attached to each other and how they are forming a common duct for the passage of different digestive juices. Now let us look at the structure of liver, how the secretion is going to be formed, in which cells the secretions are going to be secreted, and how they are transferring out of the liver. So this liver, as I said you before, that it is the largest exocrine gland, and its secretion is bile, we all know, and bile is a secretion that do not contain any enzymes. We cannot find any enzymes in the bile juice. The weight of the liver is 1.6 kg. It is made up of 4 lobes. 2 main lobes are found, right hepatic lobe and left hepatic lobe, and 2 smaller lobes are present, quadrate and cubital. And gallbladder is a balloon-like organ which is present in one of the smaller lobe of the liver, and that is in the quadrate. So gallbladder is found in the region quadrate of the liver. Now, when you see the structure of a lobe of the liver, then you see the arrangement of different cells that secretes the juice called bile juice is showing this kind of structure, and this structure is covered by some material. It is membraned by some material, and that membrane is called as Gleason's capsule. So this Gleason's capsule is present as the outer covering, and you see the presence of different cells. Firstly, in the middle, you can see a central vein is present, and to this central vein, different plates of cells are present. They are attaching the central vein. So the cells are called as hepatocytes. The cells which are present in the liver, they are called as hepatocytes, and they are rich in glycogen. And when you see the arrangement of these cells, then they are arranged in a chain-like manner, in a plate-like manner. And this chain of cells is called as hepatic cord. Cord is a word which is used for a synonym for a chain. So hepatic cord, we can say. The chain of cells, it can be called as the hepatic cord. And besides this, in the middle of hepatic cord, some space is present which is filled with fluid, and this space is called as sinusoid. In this space, some more cells are present, and we call that cells as Kupffer cells. Kupffer cells are the phagocytes. So phagocytes means they engulf foreign particles. If any foreign material enters in the lobe of our liver, then Kupffer cells are responsible to engulf that foreign particles. So we can call these Kupffer cells as phagocytes. This is about the different cells that are present in the lobe of a liver. At the end of this hepatic cord, one duct is present, and that duct is called as bile duct. This bile duct collects the bile which is secreted by the liver cells. And you can see the presence of hepatic artery and hepatic portal vein also in this structure that carries the liver secretion. Now, I'll elaborate this diagram. When you see the large diagram of this hepatic cord, then it would be like this. In between the cells, some space is present, this space. And this space is called as hepatic canaliculi. We call the space as hepatic canaliculi. So, whatever the secretions are secreted by these hepatocytes, it is taken up by this hepatic canaliculi, and all the hepatic canaliculi transfer this secretion to a common duct that is present at the upper portion, which we call as bile duct. So before the bile enters into the bile duct, it is taken by this duct, and this duct is called as hepatic duct. So firstly, the hepatocyte secretes the material, secretes the digestive juice called bile. This bile is collected by hepatic canaliculi, and from all the hepatic canaliculi, the bile is collected by a duct called hepatic duct, and from the hepatic duct, it is going to be transferred to the main duct which we call bile duct. So in this way, the bile secretion takes place in the different liver cells, and they are going to be transferred through the different ducts and help in the process of digestion. So this is how the different cells are arranged in the liver, and they are responsible to secrete the digestive juice called bile, which do not contain any enzymes in it. In the next session, we will be learning about the different functions of the largest exocrine gland called liver. So that's all for today.